Today's guest speaker, Dr. Kirti Dube from Sevadal Mahila Mahavidyalaya, Nagpur. Organizing committee members, Dr. Vidya Kathoke Madam, Dr. Pohekar, Diksha Zhare, and all my dear students. I welcome all of you in this 3 days webinar series, especially organized for the students of microbiology to boost up their knowledge and to create interest in this subject. Friends, the topics that we have selected are very much current and vital, especially keeping in view the corona pandemic situation in mind. And we thought that students must be acquainted to these topics so that their doubts will be cleared and they will get correct information and knowledge about these topics. You must have gone through the flyer, and the 3 days topics that we have selected is, first one is learning perspectives in research in microbiology, which will be, this guest lecture will be delivered by Dr. Kirti Dube. The second one is PCR technology and how it is used in molecular diagnostic. That will be dealt by Dr. Seema Nimbate. And the third one is immunology of hypersensitivity. This topic will be delivered by Dr. Vaishali Pimpre. Now, without taking much of your time, we will move on to our guest lecture today's. But before that, I will introduce the guest speaker to the audience. Actually, Dr. Peshwe was to address the students, but due to his ill health, he has conveyed his best wishes to this program. So he has sent his regret that he wanted to interact with the students, but due to some unavoidable circumstances, he is not available. So thank you, sir, for that. Now, Dr. Kirti Dube is Associate Professor in Sevadal Mahila Mahavidyalaya. She is having 25 years of research experience and 15 years of teaching experience. Before joining this college, Madam worked in NERI as a senior research fellow under the guidance of Deputy Director Dr. Asha Zuwarkar, a well-known name in environmental microbiology. Madam has published 51 research papers in the journals of repute with high impact factor. She has completed 2 major research projects which received UGC funding, and she also has completed 1 consultancy project of Government of India, Department of Forestry. She was awarded 3 patents by Government of India. Her area of specialization is biosurfactant technology and bioremediation. She is the recipient of coveted 2 prizes. One is Distinguished Scientist Award, which was conferred upon her in international conference. And second one is Sanvik India Gender Award, and she received cash prize of ₹5 lakh for this. She is the nodal officer of BVOC courses in the college, which are run by under the umbrella of RTM Nagpur University. Madam has co-authored 3 books and contributed some chapters in renowned books. She is member of Board of Studies in Microbiology, and she is also on the editorial board of few reputed journals. And she also is a referee for very standard research journals. She has visited many countries like Singapore, Hong Kong, Lund University, and presented her research work there. She is an ardent researcher and a great teacher. I welcome you, madam, in today's webinar series.
Thank you so much, madam. Thank you so much for your kind words and appreciation. I owe you a lot. Thank you. So shall I continue with my presentation?
Yes, you can share your screen, madam. Yes.
Yeah.
Ruma, madam, can you please help her to share the screen? Hello?
Yes,
Ruma,
ma'am.
madam.
Yes, ma'am,
Yeah.
you can share your screen.
It is showing a lot of buffering here actually. A lot of buffering is there.
Okay.
So,
Okay. Students, please bear with us because this technical problem always arises when there is an online lecture. So you be there.
Archana Kulkarni for giving me an opportunity to talk upon the topic, research perspectives in microbiology. So it is really a great honor, and I thank you, the organizers, the management, and the principal sir of Dharampet Science College for giving me an opportunity for this presentation. So I would like to first explain what is actually the research is. According to WHO, the research, it is a quest for knowledge through diligent research or investigation or experimentation that is aimed at the discovery and interpretation of new knowledge. It is very clear that research is nothing but gaining of new knowledge from the previous existing knowledge which is available, because we would like to have lots of proofs to prove that whatever said earlier is true in the existing scenario also. So we have to prove it by various models of experimentations so that the output which we get, it is nothing but the research. So first of all, dear students and all the colleagues and the listeners, I would like to say that research and scientific methods may be considered as a course of critical inquiry, leading to discovery of facts or information which increases our understanding for the betterment of the human being in terms of health and the rectification of the diseases. So being microbiologist, I would like to pay a lot of focus on how the product of the microbial origin can be increased or can be put into the industrial scale, what I had done in my research. I'll be giving some of the quotations or some of the points which I had done along with the presentation, what the basic presentation about the research is. So meaning of research, that is, research is an endeavor to discover answers to intellectual and practical problems through the application of scientific methods. It is a systematized effort to gain new knowledge, like Redman Mori, Fred Perlinger.
Has stopped. Your screen sharing, your slide is not changing.
Okay.
Madam, please start your presentation actually. You have not started it.
Start
What?
Your presentation.
Screen sharing is not available. Screen is
now
not shared.
Now only the first slide was showing. Now the second one we can see.
See here. Now this meaning slide, which is having the heading meaning, is it visible?
Yes, ma'am.
Okay. So research is an endeavor to discover the answers to the intellectual. And this is said by Redman and Mori. According to Fred Karlinger, it is an organized inquiry designed and carried out to provide information for solving a problem. So according to Francis Remuel, it is a careful inquiry or the examination to discover new information or relationship and to expand and to verify existing knowledge. So if whatever existing knowledge is there, as we say that a lot of Ayurvedic drugs are there, but we don't have the proof whether they are really having the potential to treat the disease. We do not have the background or the research behind it. So we do not believe it. But once we have got the research and the data available, then we can say that yes, it is really worth having Ayurvedic drugs for our disease cure. Okay, so it is essentially an investigation or the recording and analysis of evidences for the purpose of gaining knowledge. So research is a systematic process of collecting and analyzing information data in order to increase our understanding of the phenomenon about which we are concerned or interested. When we talk about the research methodology, research is an art of scientific investigation. It is a movement from known to unknown, because whatever is known, we have to prove it. Yes, it has happened or it has occurred. So by the virtue of the experimentations, we have to find out the unknown aspects which we do not know. It is a systematic method of finding solutions to a problem. Search for knowledge through objective. First, we have to design an objective on which we want to work on the research. And then we have to perform experiments. And through results of those experiments, we can gain the knowledge. Yes, this can happen or this cannot happen. So it is a pursuit of truth with the help of study, observation, comparison, and experimentation. When I talk about categories of research, there are two different types of research in which the microbiologists are generally interested. They are two different types. They are basic and the applied research. In case of empirical research and theoretical research, it is it is in the purview of the mathematics or the arts and the social science faculties. They or the students, they undergo or they do this type of empirical and the theoretical research. Through the theories, they will prove what they want to have the objectives in their coursework. So when we talk about a microbiology research work, there two different types of research is possible. It is the basic research and the applied research. Then I would like to tell you about what is the difference between the basic and the applied research. The basic or the pure research, it is generally considered to involve the search for knowledge without defined goal of the utility or the specific purpose. But when we talk about applied research, we know that we want to use, for example, the bacteria for the production of some amino acid, okay, or some enzyme. Then we should know how we can scale up the process. We know what we want actually. But in case of applied research, when a microbiologist whose expertise is in identification of the pathogenic microorganisms may conduct research in vitro in the laboratory on bacterial reproduction, growth, endotoxin production without any direct application to clinical aspects. But he will go on doing research on the characterization of the organism, which is the basic research. But when we are using that particular organism for the production of some industrial compound or for some industrial purpose or for some application, then it becomes an applied research. Is it clear, dear students? So probably the researcher is interested in monitoring endotoxin produced over the life cycle of the bacteria to determine its pathogenicity. So some production aspect, if we are dealing with, for example, endotoxin or some biosurfactants or some enzymes. So these are the basic characters of the organism which we study. But we want to apply, or when we want to use these research parameters, it varies. There we have to optimize the several conditions where we can get the maximum production of those compounds. So there we have to go to pilot scale and then to commercial scale. Okay. Applied research, it is a problem oriented and is directly towards a defined and purposeful end. Like I want to increase the production of some enzymes. So I will, for the commercial scale production. So that is the purpose. That is the purpose of that particular enzyme. We know the purpose of that particular enzyme. It has a lot of application in several industries. So we are doing the things in a purposeful manner. Okay. So, for example, an oral health researcher investigating relationship between periodontal diseases and myocardial infarction may use the findings of microbiologists as a theoretical framework to test the relationship between the bacterial endotoxin periodontal disease and the myocardial infarction in animal and then in human subject. So if these two parameters, if the periodontal disease, if it is related to myocardial infarction, if we want to study the effect of this disease on the myocardial infarction, then what we are supposed to do, we are supposed to determine how this disease is harmful for the human beings, and it is giving the medical problem of myocardial infarction. So if I want to study a particular parameter for the benefit or for the purposeful work, that is, I want to determine whether that endotoxin is having an effect on the myocardial infarction disease or not. So if I want to analyze this, then I will use an applied research aspect. Okay, because I know I have a purpose to determine that the effect of endotoxin on myocardial infarction. The methods, techniques that are used for conducting research are termed as research methods or research techniques. The methods which we use for conducting research, by what method you are using the research, what techniques you are using in your research, that if you are using for the research, and it can be of three types, those techniques can be of three types. First, you have to collect the data of the results what you have obtained. Then you have to apply the statistical techniques to verify the data, whether they are a correct data or they are having some variation. Okay, how much it is determine the variation in the results, and those methods which are used to evaluate the accuracy of the results. So statistical methods are also used to evaluate the accuracy of the results that you obtain during your research work. Okay, so this will later on in some details. Okay, so when you are doing your research, at your post-graduation level, there you have to submit a dissertation, and this dissertation is nothing but a mini thesis of a small part of the work that you do in your PG level, in your PG course. That means you have, for the 6 months, you have to do your research in a particular topic, and then you have to compile the whole what you have obtained during your PG work. And this, it is nothing but what dissertation means. Dissertation is a mini thesis, as I told you. It is nothing but the academic activity laid down by the university for the partial fulfillment of the post-graduation degree. So if you have the research work carried out during your post-graduation level and you have formed a booklet, then such booklet which is having a flow of introduction, review of, then result and discussion about what results you have got, and you have to relate the results with the available information in the literature. Then you have to find what summary and the conclusions you have drawn out of those results and discussion. And this becomes a small thesis. And it is an exercise to train the PG students to plan, execute, evaluate, write and report of a scientific manner. Okay, it is an in-depth study of a particular topic which contributes new information and knowledge in the field. But how you will find out whether that knowledge you have gained is new or not? This you have to do on the basis of the literature survey. First, you have to do the literature survey. Then you have to find out the problem which is still existing on a particular topic. And then you have to select that particular topic for your thesis or for your dissertation. A formal 10 lengthy treatise or the book or the writing of some particular subject, one containing a methodological discussion and exposition of principles of the subject or discourse, especially one written by the candidate for the master's degree. So what should be each chapter of the dissertation that I will be dealing it with the thesis part, because the dissertation and the thesis part, the chapter wise distribution is the same, okay, of what work is to be done according to the chapter. I will be explaining you in the coming slides. What thesis is, according to Oxford English Dictionary, it is a proposition laid down as a theme to be discussed and proved. So as theme to be discussed and proved, because scientific methods or scientific or research, it all needs the proofs. Okay, what proofs you want to establish, what you have got to establish what earlier people have said. So a discussion to maintain and prove a thesis, especially written or delivered by the candidate for university degree, that is PhD. For the PhD degree, the research booklet, what a candidate prepares, which has the 5 main chapters, as we'll be discussing now in the coming slide, it is nothing but a thesis, okay? The dissertation in a bigger overall scenario, like containing lot of integrated aspects of a particular topic. When you discuss, when you work on it, it becomes a thesis. Okay. Now, the thesis chapter. This thesis chapter will also be the same for the dissertation chapters. It will have introduction. First, in the introduction, you have to introduce the topic, and the topic is to be introduced in such a way that you have to have a prior art of that particular topic, and then you have to say that since so and so work has not been done, so you are intending to present that work, and you have to you want to present this work in this particular thesis. Then comes the review of literature, which I will deal it in details in the coming slide. Why it is so important? It is the first priority to have the review of literature on the available topic that is given to you or assigned to you by your guide. Then comes the methodology. What methodology you should have to use. One method may be applicable for your work. It may not be applicable for other person to work on it. So that person has to modify the work. So we write modified method of so-and-so scientist. So result and discussion, it is very important aspect. That also will be. Then comes lastly the summary and the conclusion of the thesis and the dissertation chapter. That is nothing but what you have found out after doing the big scientific exploration. So problem selection criteria. Whenever you select a particular topic for your PhD or for your dissertation, it should have first the uniqueness. Then how you know that uniqueness is there in your topic? First, we have to do lot of literature survey, thorough literature survey. So what uniqueness, how you will find out? For this, finding from the research should contribute to the body of knowledge already in existence, not merely duplicating existing study. What uniqueness, how you will get? You should not duplicate the work which has already been done, but work which is still to be remain to be done, which nobody has reported, which nobody has done, that work you have to select as your PhD topic. Okay. So, however, to pursue a study similar to one already in existence, but change the method used, or modify the design, or use a different sample, or choose to perform different statistical analysis. So what already people have done, it is to work on it, it is of no use, because you will not get any publication, you will not get any patents on it. But when you modify something or some earlier reported techniques, so a better technique might evolve. So by which you can perform your experiment. You can modify the design of your experimentation. You can modify the media, what you can use for the growth of the organism. You can modify the nitrogen concentration, particular optimum nitrogen source for the growth of the organism. You can choose new nitrogen source or any ingredient of the growth media, so that you get a different result or different observation what others have not observed. So use a different sample. Sample can also vary. When I have worked on biosurfactants, at that time, people were working on the synthetic media for production of biosurfactant. But what I thought, why not to use some no-cost substrate for the production of biosurfactants? So what I did, I started screening different industrial waste. If these waste can be used for biosurfactant production, such waste is of no cost. And when we are recovering biosurfactant from such waste after growing the biosurfactant producing organism in those waste, because waste as such, you all know, it is nothing but the combination of different forms of organic carbon, nitrogen, phosphorus, sulfur, potash, lot of micronutrients available in them, along with some toxic compounds that is also there. So you have to find out such an organism which can grow on those industrial waste and produce your product, which is biosurfactant. So it is not necessary that people, whatever they have used, you should also use the same method, your same medium for the production. You have to modify your work so that it becomes a novel work. So even the analytical parameters, what you use on analytical methods, what you use, you can modify them. One thing I would like to quote, what people have used for mutant isolation was the pure synthetic medium, and that too nutritionally rich medium is required for isolation of mutants of organism which we want to have, so that the larger production or higher production of some compound you will get. In my case, in my work, what I had done, I had not used any pure chemical based media, high nutrient rich media. What I used was the whey waste, which is a very good source of high nutrient content in terms of proteins, okay, proteins, then phosphorus, nitrogen, everything along with vitamins. So in that, I grown the mutants, and mutants were isolated in whey medium, in whey agar. So that is a novel work. So that also fetched me some very good publication. So I wanted to tell you, dear students, don't go for the already available methods, already available designs, or the protocols, what you follow for any work, what you do in the research. Have some novelty in it, okay? Now, but how you will select that this particular objective, what you have selected for your research, it is a novel work. How you have determined that nobody has done. For this, so we have to stuck to a particular topic. So we have to stick to a particular area while finding the available literature. First, we know what the problem we have. You. Actually, what is literature review? It is nothing but the critical element of the research, and it is the database compilation of the research that has been published on a topic by recognized scholars and researchers. So we have to study what earlier the researchers have already established through their experimentation, what results they have got, and according to that, we have to find out what they have not done and what you can do so that you can have your good research, that is research of new aspects that you want to search. Why we need to do and write research literature review? It is to acquire and demonstrate knowledge in a particular area. But first we have to demonstrate that only this much available information is there on a particular topic, and to convey that the knowledge and the ideas have been established on specific topics, and what things the other researchers have not attempted, you can do it. This you can write in your review literature that since earlier so-and-so work has not been reported, therefore you have taken up these aspects to be performed through experimentation in your research. So it is also the theoretical framework and the methodological focus. So this review of literature also tells you about how the earlier researchers have used the methodologies and the designs of their experimental models and how they have carried out their work. Now, once you have done the review of literature and you have found out that these aspects have not been taken up by any of the researcher, then you will have the objectives defined. And once the objectives have been defined, that these are the aspects you want to take up in your research, you have to have a hypothesis that is nothing but an educated guess whether you can apply the hypothesis for all particular research work. Yes, we have to apply hypothesis, that is educated guess, or that you can assume or presume that. So these can be the output of your research. So if you want to have particular result and of a particular topic, then you can also have the hypothesis designed. It is nothing but the attempt to explain the nature of the relationship between the variables identified in the problem. It is an attempt to suggest a possible answer to a problem based on the available facts or the information that the researcher already knows. So hypothesis, it is based on what available information is. Upon that only you can frame an hypothesis. And it gives the direction to the study. Without hypothesis, you cannot perform your work. Hypothetically, you have to assume that you have to have this set of design so that you can have the result. Okay, so without hypothesis, you cannot have a particular direction that from where you have to start your work and how long you have to work on it. Okay, what are the different aspects you have to cover up in your research work. It gives the evidence that some background reading in the area has been carried out. So for having a hypothesis, you have to have a background. Without background, and this background comes from the review of literature. Okay, based on the review of literature, you have to have an hypothesis on the objectives that you have selected for your work, and then the hypothesis written after the review of the literature has been done and the problem has been selected for the study. So the objectives have to be hypothetical. The hypothesis has to be laid down based on the objectives, and so that you get the proper direction from where you have to start your experimentation. It is a prerequisite or the academic activity of acquiring a master's degree or the doctoral, and to find out the truth which is hidden and which has not been discovered yet. So hypothesis, it is the way by which you can find out what is hidden. That means what is not known and what is to be discovered yet. And to contribute a new knowledge to the existing one, the scientific knowledge development is very important. And through hypothesis, you can have, or hypothesis can lead to the knowledge development. A profession to conduct research in various aspects and to solve an existing health problems, as in now presently in the COVID situation, it is occurring. And even for the thrill and enjoyment of the discovering a new fact. So it is very important to have a hypothesis. Then, what is result and discussion? What is result and discussion of the thesis? What you have a very important chapter, that is result and discussion, and how you write this particular chapter of your thesis. This is a chapter of the thesis that present the finding and the outcomes of the research and the reasons for that particular result in the form of text, figures, graphs, and charts. So in the result and discussion part, you have to compile your data, whatever data you have obtained or whatever results you have obtained, that in the form of text, the written text. And then in the written text, you have to support your findings with the help of figures and different graphical representation, and even charts also you can prepare and you can add in your result and discussion part. Now, the results tells about the outcome or the findings of the result of the research study. It presents the data and the findings and ordered or analyzed in ways justified earlier. That is through the methodology. You will be proper use of methodology or proper use of methods. You will be performing your experiment. You will get your results, but how you represent your result in the result and discussion part, it is very important. Whether the result will be whether the result presented as graph or graphical representation will be more revealing about the results, or in the tabular form it will be okay. So like that, you have to have the guidance of your research guide, and research guide will help you to analyze, and you will also learn in due course of time that how you have to represent your results best, so that the outcome of your study is pictured, or it is in front of the viewers or the readers at a glance. Okay, so the past tense is always used when you write the text of the result. You have to use past tense. That is, from the above observation, it was observed that, or from the results presented in table so and so, it was observed that. So all the past tense has to be used in the sentences when you write your result, usually. Then it describes the findings in the simple way with the help of data. So the supporting data should be there in the form of either graph, or in the form of table, or in the form of charts, so that the supporting data you have to always compile along with the results. Okay, that is the text material. Then figures, tables appear here in the result and discussion chapter of your thesis. Then what do you mean by discussion? Results, okay, you know that whatever experiments you have performed, you will be writing the results. Okay, the effect of, suppose, nitrogen concentration was very much pronounced when biosurfactant was found to was taken into consideration. So which nitrogen source was the optimum source, and at what concentration it gave the maximum production? So at that time, if you want to have the pictorial view in the form of graphical representation, then it will be very good. But how the results, what you have got on this particular nitrogen source and the concentration of the nitrogen source on biosurfactant production by the other scientists, by the other researchers, and how your result is comparable with their results. It is not necessary that the same yield of biosurfactant you will get at the concentration what you have used. It may be because of the variation in the strain or the organism you have used. So every time you have to support your data that you have got this result, whereas the earlier reported observations or the results were this by so-and-so's researcher. So what does discussion deals with? It is the result of the data from the experiment means to us is described in the discussion. That means what does the result of the data from the experiment mean to us is described in the discussion. The function of discussion is to interpret the result in the light of what was already known about the subject. That means what is known about a subject, that is, for example, what I had quoted, that what already has been reported on biosurfactant production if different sources of nitrogen were used as the nitrogen sources, like ammonium chloride, ammonium sulfate, ammonium phosphate, sodium nitrate. So if these were some of the nitrogen sources used for biosurfactant production at 2% concentration, for example, how other researchers have got their result? What was the yield of biosurfactant? And what you have got when you are doing that work with those nitrogen sources. So explain the new understanding of the problem of taking result into consideration. Explain how the results answer the question under the study. So it is nothing but you have to discuss what results you have got in comparison to the results reported by other scientists. So when I talk about result and discussion, let us revise what result is. It is nothing but the core of the research paper or the thesis. Okay, it is the core of the, or it is the main part of the thesis or the dissertation or any research paper. The data generated by doing research after validation and verification is written. After validation and verification is written. Okay, the data is validated, it is verified, okay, it is statistically verified, and then it is validated also through statistical packages like SPSS package, ANOVA package, that I will deal it further. Okay, the results are important in answering what results you have got when you have chosen the different objectives for your research work. Findings of the research, it is nothing but the result of the experimentation or the results of your research. Then discussion, it is nothing but stating the cause of a particular result, why this type of result has occurred or happened, or you have got the result, why you have got this result and why you have not got the other aspects signifying the results. So discuss the meaning of the result, stating clearly whether their significance is, then compare the results with the theoretical expectations. If the theoretical expectations are also considered when you discuss the results with the other scientists. Okay, so this is very important. Dear students, the main aspect when you have chosen your research work, first you have to identify the problem that you have taken up, and then you have to do the literature survey, then you have to plan a research design, and then you have to determine the sample size, and that means how many times you will be taking the observations or you will be doing the analysis. Okay, so you have to write a protocol by which or by what method you will be finding the analysis or you will be performing the analysis. Then you have to have an ethical committee approval also. This is very important when you do the animal studies. Okay, when I want to study any effect of endotoxin on some animals, for example, guinea pig, then I will have to have the approval from the ethical committee, that is animal ethical committee, so that I can get the approval for the trial. Okay, so then after that, and if I want to study if the biosurfactants what I have produced, it is safe for cosmetic purpose, if it is safe for cosmetic purpose, so then I have to do some animal testing. Okay, for this I will be needing the ethical permission or the ethical committee's approval, so that I can give the sample for analysis, and whatever results I get, that will be useful for the application of biosurfactant for the cosmetic purpose. Without animal testing, the products produced by microorganisms cannot be used for human application. Okay, now then if I want to have the commercial production, then the pilot study and the main study is very important. I have to undergo several pilot scale studies. For this, I'll be needing funding from different commercial sectors or the industries who are interested in the product. Then this product, if anybody is, if any industry is interested, then it will give us the funding, so that I will have a pilot plant at that particular industry and produce the biosurfactant. And this is very important, so that the product is commercialized. If it is having cosmetic application, then it has to be produced in pilot scale and it has to be commercialized. And then presentation of analysis of result. Then I should present whatever result I have got with a proper, clear, pictorial or graphical representation or the tabular form. Okay, so the hypothesis testing is also very important, as we discussed earlier. Then generation of interpretation, then report writing is very important, and lastly the publication. So this is the general scheme of research which is represented here, that how it ends up in publication, and which is very much important for the scientific arena. Then the research process, the first step is we have to define a problem and the research objectives. Then the second step is the design the research with the plan of collecting data. Then third step is implementation of the research, collect, prepare and analyze data. Then fourth step is produce results, interpret, deduce, inference and report the findings. So these are in the steps that you have to perform the research on a particular topic. So these are the 4 steps. As we have already discussed this earlier also. Research design, when we want to design our research, then a specification of the method and the procedures for obtaining information is needed. We have to specify what methods you will be using. If you want to analyze a particular parameter, then what procedures you have to use, what you have to standardize, so that you get the proper result. Okay, so a plan of organizing framework for conducting the study and collecting data serves as the blueprint for detailed procedure and rationales of the research project, an essential part of the research methodology. It is very much important that you follow a particular design, and it should be discussed with the guide, and the student should organize the framework of conducting the study. This is very important in consultation with the guide. The importance of research design are such as it serves as a foundation to formulate and guide the research study, supports in better planning, execution of the research methodology, useful in estimation of probable research errors and handling strategies. See, if you don't have a research design proper, it will lead to a lot of errors, and errors will be enormous when you take up the statistical analysis. So with minimum of error, you have to start, because human error amounts to 100-folds when you do your work. So as you minimize the human error, the results will be more accurate. Okay, so maintains necessary control over the contents of the study. Otherwise, we have to end up in doing a lot of analytical work with less of results, because every time it is not possible that you will get results from whatever you have designed the framework of the research. Makes the study more systematic and effective. If you have a proper design of the methodology, what you will be using to perform your research, then your work will be more systematic and it will come out with a good result. Okay, now research design, it will ask several questions. Before these questions come in mind, you cannot design a proper research protocol. What study is all about? why the study is being carried out. First, you have to analyze why you want to carry out this particular objective as one of the important study. Why, where will this study be carried out? What tools and techniques of data collection you will be using? How will the issue of validity and reliability will be addressed? Because validity and the reliability of the results is very important when you have to have results. Therefore, you have to perform your experiments in 3 sets. First set, second set, and third set. Both will give you some variation in the results. So that variation and how much the variation is deviating, it can be determined with the help of statistical analysis. So at least 3 sets you always have to plan before you carry out your work. If I want to work on how much is the biosurfactant production from whey waste, then I have to have 3 sets of 3 flasks over a period of 5 days. That means 15 flasks I should have. So that over a period of 15 days, I will be having daily 3 flasks for analysis. How much is the production of biosurfactant by a particular organism in whey waste? So like that, I will get the results. It may not be necessary that the same result or the same 1.05 grams I will get in all the 3 flasks of the yield of biosurfactant. So, but at least I will have the data of the 3 set. If one set goes missing or some happens, if flask breaks, then I at least have 2 sets with me. So like that, you will not miss anything, but you will have an extra data so that your result will be more accurately predicted or validated, and it will be more reliable. So what type of data are required that you have to determine and you have to address with the help of your guide. What will be the sample design? What techniques of data collection you will be using? What techniques of data collection? Suppose I want to analyze biomass production in whey waste or biosurfactant producing organism over a period of 5 days. Then what method of biomass estimation I'll be using? Whether I'll be estimating the total protein content of the cell biomass, or I'll be using the cell dry weight, or I'll be using the wet weight count. So it all depends upon what is the actual requirement of at that particular moment. What is feasible in your laboratory? How best you can do, what best technique you can use. So how will the data be analyzed and interpreted? So that again, you have to consult with your guide, okay? How data can be analyzed and interpreted. So presentation of data or presentation of your result, whatever you get, it can be in the form of descriptive test. It can be in the form of figures, like graphs, pictures, diagrams, charts, histograms, then box plots and other drawings. Okay. Then presentation of data, tables and figures. Figures, it may also contain photographs, drawing graphs, and flow diagrams. Flow sheets are often used to present details, whereas the narrative section of the result tends to be used to present the general findings. The narrative section, that is the text part, it will be there in your result portion of your thesis or the manuscript or your dissertation, along with the tables and the figures. The numerical data usually be presented more effectively in tables and graphs than in the text. The numerical data, like if I want to determine what is the effect of, suppose, glucose on biosurfactant production. So I will have the data that will having for 5 days, the amount of biosurfactant produced in some grams for 5 days, and I will be plotting a graph. Okay, so for 5 days, I will be having on my x-axis, and on the y-axis I'll be having the grams of biosurfactant produced. So I get the data in the form of graph. Okay, that graph I had made it from the numerical data. So the numerical data can also be presented in the form of graph, which will be more revealing, or which will be more perfect for the presentation. Okay, the order of presentation of the result should be either chronologically to correspond with the methods, or from the most of the least important. So this, it all depends on what your guide prefers. Okay, and your tells you to follow. So if this is the table form, so the table is an organized set of data elements, that is values, using a model of vertical columns. These are the vertical columns, and on top you have to write down the different parameters. Okay, so for this class interval parameter, this is the numerical data. For the frequency, it is this. For cumulative frequency, it is this. So like that, you can put, or you can add the data, what numerical data you have got, in the form of tables, which is nothing but the table having different columns, and that is vertical columns, and the rows which are horizontal. Okay, the cell is the unit of the table. This particular single cell of this table is nothing but a one unit of the table. So it can be either a row, or it can be either a column intersect. See, this row and this column intersect here. Okay, so this is nothing but the form of table that you can use, and you can put your numerical values here in the table form for representing your result. Now, even the charts can also be prepared. Okay, charts can also be prepared. So like this, you can prepare chart through the computer. Okay, a chart is a graphical representation of the data, numerical data, and it is represented by symbols such as bar in the bar charts, line in the line charts, or the slice or the pie chart. So line diagram also you can have of a graph. You can have the bar diagram like this, or you can have a pie chart also. Okay, the chart represents the tabular numerical data. The table, the data, what you have inserted, or what you have prepared in the tabular form, can be converted into the chart form, can be converted into the graphical representation. Okay, so that looks much better. You can see here the variation in the parameters. Okay, so series 1, 2, and 3, these are nothing but the different parameters, and the categories here you can mention as per your results. Okay, so these are the graphics. Through graphics also, this is nothing but the map just shown, that also you can insert. And for example, the graphic designs like photographs, drawing, graphs, then the diagrams, symbols, and maps can also be inserted in the result and discussion part of your thesis. Now, data interpretation is very important when you prepare your data, or when you want to represent your data. Before that, you have to interpret the data. Okay, what is the result outcome of the data? The analysis of the caution of the effect by a factor, interpretation in multi-causal factors and multi-effects, statistical association, and the application of statistics in interpreting data. So in interpreting data, you have to determine what is the parameter which affects the production of some value-added product. So that parameter which affects the production is the crucial parameter. So that the data of that particular parameter you have to first pick up. Okay, then you have to see that parameter is being influenced by what different types of factors, so that it will affect the production. Even production in how much amount you are getting, that you can statistically validate. And therefore you have to apply statistics in interpreting the data. Application of statistics. Statistical difference is the function of magnitude of difference, variability of difference, and the sample size. You should, if you want to know what is the difference between the results what you have got in the 3 sets of the flask on biosurfactant yield. So how much is the difference you are getting in the yield in the 3 different flasks? There you have to apply statistics. What is the magnitude of the difference? How much is the difference? How much is the variation? And what is the sample size? That means how many flasks you have taken at a time to interpret this data. That means I've taken 3 flasks. Some they take 5 flasks or set of 7 flasks. So more the number of flasks you take, more accurate becomes your data. Okay, more accurate becomes your result. Now, statistical measures for analysis. They are the measures for central tendency. They are what? Different parameters. By virtue of mean, mode, and median, you can determine what is the central tendency of the result. Then measures of dispersion. You can use standard deviation and variance. Then measurement of relationship, that is correlation, regression analysis, and the t-test and the z-test. So these are some of the analytical parameters for statistical analysis of your result. Okay, so mainly we use standard deviation, standard error, then t-test, and the regression analysis. So these are the some statistical measures that you can always use and you should use to analyze your data. There are statistical packages also available. In those statistical packages, you just have to feed your results. Okay, several columns you have to fill up with the results or the data what you have got for the 3 different flasks or 5 different flask experiments. Okay, that is in triplicate, in duplicate, in quadruplet, like that. How many readings you have for a single set of experiments? So those readings, all you have to feed in the ANOVA package. That is one of the statistical package. It is generally used and it is must for the researcher to use. Then you have to validate your data. You have to validate your data. What you have got is a perfect, or it is the data, or it is so you have to validate whatever you have got the results. Okay, without validation, how you will do the validation of your results? By way of publication, and performance is computed from the result. Appropriate conclusion is drawn from the result. Complete documentation has to be made. What results you have got from the objectives, what you have defined for your work. So this is all summarized. It is prepared in the form of booklet or in the form of manuscript, and that now you must publish it, so that after publication or after approval of the publication, That means your data, what you have got, it is validated from the scientific community. Okay, and it is peer reviewed. That means it is after a peer thorough review process, your work has been validated. Okay, and it is found to be correct. So moving for publishing, a document or report findings, you should always publish it because you want to validate what results you have got of your experiment. Without publication, nobody will validate your data. Okay, a way to verify your findings, it is nothing but a way to verify your findings. A contribution to advancement of science, it will also lead to advancement of science when you publish your data. Mode of knowledge sharing, that means your data you are sharing with the masses or with the large community of the scientific community, and you are sharing your data with the large masses. That means you are sharing the knowledge. That is acknowledgement of the intellectual value. It is a document of intellectual value. That means through intellectual or through your research, the document has been prepared. That is a proof of author of scientific creditability. If you want to give credit to a scientific work, then you have to publish your work. Then only the people will give you credit that yes, you have the acumen or intellectual ability to present your data, to publish your data. Otherwise, your data has no value. Types of article writing. There are different types of articles you can have, like popular articles, then research proposals, reports in the form of theses and the dissertations, then conference proceeding articles. You can also publish in conferences, and you can also present your data in different conferences, national and international conferences, so that the masses are well aware of what work you are doing, what is the importance of your work, and thus the creditability of your work. And the journals, there are two different types of journals, that is non-scientific citation index journals and the scientific citation index journals with impact factor. These things will be taken up now, dear students. Publication checklist, establish the list of the journals that you should know in what journals that you should publish your paper. First, it should be science citation index journals with high impact factor. Then you have to develop a publication plan that you want to publish your data. Then what data is actually having a credit to be published, you have to plan for that. What you have to publish, how you will publish, how well you can write your result, how well you can discuss your result, how well you can have a conclusion of your particular work done. Organize writing retreats. You have to organize writing retreats. That means you have to write. By writing the papers, first you will learn, and then you will become a master in writing. So of course, but before submitting the paper for publication, you should thoroughly give it for editing, or you should review the paper so that publication is not hindered. Okay, so editing by an expert, your guide, and the guide may give to their own peer reviewers. Okay, so that whatever work you have presented in the form of manuscript, it is the best one, and nobody can challenge the publication. When you are publishing a new aspect, always it is published. So mind well, dear students. Then research planning. obtaining results, organizing results, planning for publication. These are the 4 important aspects when you write your thesis or when you write your paper for publication. So first, research planning, obtaining results, then organizing results, then planning for publication. These are the 4 important aspects that you should always keep in mind. That means you always end up your research in publication. Okay. So paper writing philosophy. A paper contains a report on new findings or a solution. Purpose is to convince the readers that your findings and solutions are important and better than the existing solutions and better than what earlier others have reported. Okay. You have got the much improvised, much better knowledge or aspects you are giving to the society. Then only the paper will be published. Then writing process flow, that is pre-submission review. It is very important. The reviewed papers before submission should be given for publication.
हेलो मैडम सॉरी टू इंटरप्ट, आपको अभी ज्यादा टाइम नहीं है, आप थोड़ा सा वाइंड अप कर दीजिए प्लीज.
Yes ma'am. Yes ma'am.
ठीक है.
Then you have to correct after reviewing. You have to correct the paper and then you have to submit it. When we are performing our research, then the performance of the research of a particular candidate, it is always determined by how many number of citations your paper has in the society, how much of your papers are indexed in Scopus, and what are the scientific citation index of your paper, and what is the impact factor of the journal in which you have published your paper. So each of these are very important, and these decides the credibility of the researcher. Okay, so about SCI journal and non-SCI journal, the SCI journals for science citation index, it is now taken up by the Thomson Scientific Society, and for non-SCI, the elsewhere publishers, the Scopus and the Science Direct. These are some of the very important publishers, and under these publishers are Taylor Francis, Springerling, Emerald, Blackwell, Synergy, etc. What is impact factor? I would like to tell you. It helps you to evaluate the scholarly worth of the journal, rank of the journal within the discipline, help you to decide where to publish your article for the maximum impact, and the evaluation of promotion, tenure, grants, and in some countries, even government funding of an institution. When you are publishing your paper in high impact factor journal, it means that more and more people will have a look on your paper. Okay. Otherwise, if you are publishing in a low impact factor or without impact factor journal, then it means that there is no observer for your paper. Okay. So if the aim of the research is to spread the intellectual creditability or intellect to the masses, and once it is not going to the masses, then there is no use of such research work. So I have to wind up. So this H index, it is also the factor which decides the creditability of the researcher. And none of these matrices are adequate compared with what should be the gold standard, that is peer reviewed. So any journal which is peer reviewed is the gold standard to decide that yes, your work has a credit, or it is a credible work that you have done if you are publishing your paper in a peer-reviewed journal. So even when you are publishing your work, if it is a novel work and if you are publishing it, before publication, you can also opt for patenting that work, okay? Once you have published the paper, you cannot have a patent on that. But before publication, if you are trying to have a patent for a particular process or for a particular product, then it will add to the creditability also. So, but one thing I would like to say is that always avoid plagiarism. That means don't copy what others have done. Don't copy what others have written in their manuscript directly. You have to have a plagiarism software where you have to put your complete work and to see that it is free of plagiarism. That is, you have not copied copied any of the data without citing the name of that particular researcher. If you are writing, it seems that you have done that work. So please don't do that. This is my request to the young budding microbiologist, and I hope that it will help you to become a very good scientist, microbiologist in near future. So with this, I would like to end, my dear students. So please give me permission to leave the dais or leave the lecture. Thank you so much.
Thank you, madam, for your wonderful talk on how to carry out research. In depth, we have given idea to my students. Thank you so much. Now I invite Dr. Vidya Kathoke, madam, to propose vote of thanks.
Thank you, madam, so much.
Over to Dr. Vidya Kathoke.
Yes, madam, yes. My visibility, I'll stop without my video, I'll speak. Okay, madam. Thank you, madam.
Thank you, ma'am, so much.
Good afternoon, and I welcome you all in the first day of webinar series. On behalf of Dharampet Science College, on behalf of Department of Microbiology, Dharampet Science College, I extend my gratitude to all for gracing today's event. I would like to thank our principal, Akhilesh Peshwe sir, for sparing his valuable time. But I think because of his ill health, he was unable to attend the function. But I must thankful for his support in providing the Zoom platform for smooth conduction of 3 days webinar series. I would like to thank our Vice Principal Madam and Head Department of Microbiology, Dr. Archana Kulkarni Madam, who always had an impetus for our institution and whose efforts makes today's event to happen in a very short time period, that is 3 days webinar series from 27 to 29 Jan 22. I must give applause for her work. Thank you very much, madam, again for your invaluable support in organizing this event. Now I would like to thank today's speaker, Dr. Kirti Dube madam of Sevadal Mahila Mahavidyalaya, Nagpur, for spending her precious time and giving such an inspiring speech on developing measures, a research perspective in microbiology. I am sure this lecture will encourage our students to pursue research in microbiology in future. There are some technical problems in between, but really, madam, it's very beautiful and very nicely you explained it to our students.
Thank you so much, ma'am. Thank you so much.
With this, definitely.
It's my honor.
Yes, madam. Thank you very much. I also extend my thanks to Mrs. Ruma Kapre madam for technical support. And last but not the least, I would like to thank all my dear colleagues and the students who attended the program in large numbers, without whose presence this program will not be successful. Thank you once again. And I declare this program over. Thank you very much.
Thank you so much, ma'am. Thank you, Kulkarni madam, so much.
Thank you one and all. And the program is over.
you very
Thank
much.
you.
Okay.
Thank you so much.
Thank you.
End करू madam?
Oh.